Preparation model paper 9 on physiological and molecular plant pathology. I'm N. Shankar Reddy, PhD plant pathology. The term elicitor was coined by Noel Keane, Roberts, Sharp, and all the above. Here in physiological plant pathology, the term elicitor was coined by Noel Keane. He is also considered as father of molecular plant pathology. The term elicitor was coined by Noel Keane. This Noel Keane is also considered as father of physiological or molecular plant pathology. The next one, complementary test used to find the DNA association with proteins. Fluoroglucinol test, Felgen's test, DNA microarray, and Woozout test. Here Woozout test we know very well that is used for the identification of presence of bacteria, especially plant bacterial diseases. We used a special test called Woozout test for the identification of a plant bacterial disease. Generally here the finding of DNA association with the proteins was identified by using a test called Filgen's test. So the answer is B. Technique used to identify the species of higher taxa such as orders and phylum. So DNA fingerprinting, barcoding, DNA microarrays and all the above. Generally DNA barcoding technique was used for the identification of new orders or new phylums. This DNA barcoding was first introduced by Paul Herbert in 2003. So where there are different kinds of ITS is general primer for fungi or bacteria, some kind of general primers will be there, along with the specific primers also available. So what they do, they use to sequence all the DNA and they will store the information in a barcodes. That is called a DNA barcoding, the first introduced by Paul Herbert in 2003. One gene, one enzyme hypothesis was conducted in the organism. So in which organism this one gene, one enzyme hypothesis was discovered or maybe conducted? Red bread mold, yeast, aspergillus and penicillium. It was actually conducted on red bread mold. One gene, one enzyme hypothesis, Beadle and Tatum. It is a very important concept at the time, but this concept was not using now because for one gene there are hundreds of enzymes are responsible. Before it is believed that only one enzyme is responsible for one gene action, all the stuff, but now it's for one gene there are hundreds of enzymes are responsible. Now this phenomenon was not using that much. And the next one is a substance which is injected into the animal body is recognized by the immune system and stimulate the production of antibodies. So we must understand something here. So inside the body, the compounds which are produced are antibodies. For the production of antibodies, we actually injected something from outside that is called antigen. So antigen we are injecting in the form of injection or maybe in the form of some other thing that actually activates the production or forces the production of antibodies. The next one, which of the following disease get affected by downward translocation of nutrients? So which disease actually get affected due to the downward translocation of nutrients? Powdery mildews, rust, cankers and wilts. So generally this cankers will get affected due to the downward translocation of nutrients. The next one is which of the pathogen requires living tissues for the growth and reproduction of pathogens? Biotrophs, pertotrophs, saprophytes, all the above. Generally, biotrophs are otherwise called as obligate parasites. They strictly requires a living host for the growth and survival. So generally, biotrophic pathogens like powdery mildews, downy mildews, rust. So these are viruses, viroids, all are biotropic pathogens are strictly obligate plant parasites. So which means they strictly requires a living host cells for the reproduction and the survival as well. Which pathogen draws nutrients from dead and decaying organic matter or decaying tissues? So biotrophs, pertotrophs, saprophytes, all the above. Generally saprophytes which live or feed on dead and decaying organic matter. Saprophytes, biotrophs as I told you, which lives on which strictly requires a living host. The next one, which one is a wide spectrum plant bacterial blight? That is a bacterial blight of rice, resistant gene isolated from South African rice, that is Oryza langistaminata. So this question was, I think it was asked in 2021 net. So which bacterial gene was isolated from this African rice? Varese langistaminata, Xanthomonas 21. It is a resistant gene which is actually isolated from the African rice, Varese langistaminata, which is especially recommended for rice bacterial leaf blight, which is caused by Xanthomonas varese, Pathover varese. Bacterial leaf streak is caused by Xanthomonas varese, Pathover varese cola. So what they have asked is uh, plant bacterial resistance gene like Xanthomonas 21 that is actually isolated from which of the following species of rice? Varese sativa, Varese glabrima, Varese langistaminata, something like that. They asked the answer is Varese langistaminata, where this Xanthomonas 21 gene is isolated, especially recommended for bacterial blight of rice, which is caused by Xanthomonas varese, Pathover varese. The next one is which of the following promoter is regularly used in a gene expression in crop plants in a genetically engineered for disease resistance. This is also important one. JRF, CAMV19, CAMV35, all the above. Actually, cauliflower mosaic virus, which is a double-stranded DNA virus, it contains two kinds of promoters, 19S promoter and a 35S promoter. Among these two types of promoter, 30S, 35S type of promoter is especially recommended for a gene expression kind of thing. So the answer is cauliflower mosaic virus 35S. So they will simply ask 35S, 19S, something like that. So the answer is 35S is the best promoter in genetic engineering. Which technique is used to produce antibodies without immunizing the virus? RT-PCR, phage display technique, PTA ELISA technique, and all the above. So among the given technique, phage display technique is used to immunize the virus without production of any kind of antibodies. So what are the common enzymes used in tagging for antibodies used in ELISA? They are asking enzyme used in ELISA indirectly. What are the common enzyme used in tagging of antibodies for ELISA, which means enzyme used in ELISA? Horseradish peroxidase, alkaline phosphatase, both A and B, or D is A alone. Here, horseradish peroxidase and alkaline phosphatase are the both enzymes used in ELISA. So if you see the substrate used in ELISA is PNPP, which means para nitrophenyl phosphate. That is the substrate used in ELISA. But the enzymes are horseradish peroxidase, alkaline phosphatase. These two are important. The next one, low pat test is used for the identification of. So low pat test is Xanthomonas xylella agrobacterium fluorescent pseudomonas or pseudomonas fluorescens. So generally this low part technique is used for the identification of fluorescent pseudomonads. It's important. L for leaf and O for something, P for some kind of things will be there. Low pat. So this is the technique tests are used for the identification of fluorescent pseudomonas or pseudomonas fluorescens. This pseudomonas fluorescens, now it's called as fluorescent pseudomonas. The next one is which one of the following enzyme is the most preferred in contrast to horseradish peroxidase in ELISA? So instead of horseradish peroxidase, what is the enzyme used in ELISA? Alkaline phosphatase, methyl esterases, para-nitrophenyl phosphate, all the above. Just now we discussed horseradish peroxidase and alkaline phosphatase. If horseradish peroxidase is not recommended or means of some cases or maybe not available, we can go with alkaline phosphatase. As I told you, this para-nitrophenyl phosphate is a substrate used in ELISA. So hope you guys know what is ELISA, enzyme linked immunosorbent assay. The technique was first developed by Wohler 1976 and Clark and Adams 1977. In most of the literature we might have seen this Clark and Adams 1977. Okay, and according to the options you guys select if Wohler will be there, but majority of the options and exams it is given that Adams and Clark 1977 developed this ELISA technique, enzyme linked immunosorbent assay. It is a most sensitive technique used for the detection of plant viruses. The next one is the necrotic and sunken ulcers like lesions. I repeat, ulcers like lesions on the stems, leaves, flowers and fruits is the characteristic symptom of scald, gummosis, blotch, anthracnose. So anthracnose can produce this ulcer kind of lesions. The two depressed kind of ulcer kind of lesions can be observed in anthracnose. It's a very important one. So this anthracnose generally produces acerulae, right? Acerulus. So the PCR based method in which antibodies used for pathogen diagnosis. In which PCR based method? In PCR based methods in which antibodies are used for pathogen diagnosis. PCR, RAPD, RFLP, ISEM. Generally in RFLP method we use antibodies for this pathogen diagnosis. Technique used for the identification of many different nematode species. I repeat, identification of many nematodes in a single sample. So if the single sample having a multiple number of nematodes, so what is the technique used for the identification? So RFLP, reverse blot, RFLP, reverse blot, reverse dot blot hybridization, RAPD, immuno-PCR. So generally this reverse dot blot hybridization can be used for the identification of many samples, many nematodes in a single samples. A non-PCR amplification procedure used for the identification of identification of a bacteria. So it is a non-based PCR or non-PCR amplification procedure. So that is actually used for the identification of bacteria. So competitive PCR, NASBA, bio-PCR and LAMP. Generally this NASBA technique is used. Salicylic acid dependent responses are more active against. So this is salicylic acid, the production of SCR, systemic acquired resistance only, where salicylic acid is produced. This salicylic acid is actually activates a systemic acquired resistance. This is systemic acquired resistance activated salicylic acid, which is active against what kind of pathogen they are acting. So biotrophs, perthotropes, saprophytes and all the above. So generally this salicylic acid systemic acquired resistance is more active against biotroph kind of pathogen. Whereas for saprotropes and other kinds of pathogen, ISR, induced systemic resistance, that is actually activated by the production of ethylene and jasmonic acid. So the salicylic acid is actually active against biotropes. One gene, one enzyme hypothesis was studied in just now. We discussed, right? Neurospora, that is red blood mold. Next one is a computer-based serial disease diagnostic system. So Epidem, Epiven, Proplast and Wages. So I think Proplant is the computer-based serials, especially recommended serial diagnosis system. So application of monoclonal antibodies in phytopathology was first used for the diagnosis of fungi, bacteria, virus and all the above. Generally, first it is used for the viral disease diagnosis. First bacterial avirulent gene. They are asking not fungal avirulent gene. They are asking a bacterial avirulent gene. So AVR6 and CF9 both are the first. No. CF Cladosporium fulvum is for fungi. Yes. But for first bacterial avirulent gene is AVR6. Okay, and it has other names also as well. I couldn't able to recall it, but AVR6 is the first bacterial avirulent gene. So this is CF6, CF9 that are fungal avirulent genes. So number of PR proteins are identified so far. So far there are 17 kinds of PR proteins are identified. So jasmonic acid or ethylene dependent response are more active against. Just now we have told systemic acid resistance is activated by production of salicylic acid, right? So jasmonic acid is activated by the presence of induced systemic resistance is activated by the production of jasmonic acid and ethylenes. So such kind of pathogens are active against. They're asking biotropes are systemic acquired resistance, right? And this jasmonic acid and ethylenes are active against necrotropes and some kind of saprophytes as well, but mostly necrotropes. And size of ELISA hole. So ELISA plate is generally contains 96 holes. So 12 into 8. So there are other holes are also available like 72, 96, 384, 5380 plus, 84 plus. I'm not sure, probably 5800 plus. So these all different kinds of holes are used for various kinds of purposes. But the most commonly used ELISA plate consists of 90 holes. 12 into 8, 96, 12 rows and 18 columns, I think so. So 96. So generally the size of this 96 holes ELISA is composed of 0.4 micrometer or microliter, not micrometer, microliter. So which of the following substance act as a elicitor in plants? Oligosaccharides, glyoproteins, glycopeptides and glycoproteins, all the above. So the answer is all the above. This oligosaccharides, glycopeptides and glycoproteins all act as a elicitors. Elicitor is the compounds that is produced by the pathogen that identified by the plant receptor. It's a kind of substances or maybe responsive molecules. Okay. The next one is which of the following substance in bacteria are involved in the production of plant diseases. I repeat, which of the following substances in bacteria involved in the production of plant diseases? Salicylic acid systemic acquired resistance that activates. Jasmonic acid is activates induces systemic resistance. There are actually resistance that helps the plant. But the question what they asked is involved in the disease production. This polysaccharides which is are produced by the bacteria which are the culprits in production of diseases in plants. The next one, phytoalexins. Phyto means plant, alexins means to ward off or protecting kind of substances. These phytoalexins are the plant protecting substances which are produced by the plant after infection, not before infection, I repeat, after infection. So that was discovered in studying the interaction with which pathogen? Coffee rust, lead blight, tomato wilt and frog spot of tobacco. Generally, that while they are working with the potato system, that is a lead blight of potato, the first phytolexin was discovered is P-satin, lead blight of potato, and P is some kind of interaction model where they are working with. So they discovered the world's first phytolexin, that is P-satin. That was discovered by Muller and Borger in the year of 1940. So the answer option given is lead blight of potato. So example of vivotoxin, I repeat vivotoxin, alternaric acid, cochleobolin or cochleobolin, fusioric acid and pyrrolidine. So this vivotoxin, I repeat, there are three kinds of toxin is there, phytotoxin, vivotoxin and pathotoxin. Among all the toxin, this pathotoxin is very dangerous. So phytotoxin best example is fusioric acid. Vivotoxin best example is phytotoxin best example is alternaric acid. Phyto means plant. So vivotoxin best example is fusioric acid. The term vivotoxin was given by Diamond and Wagner 1953. But the concept was given the concept of this vivotoxin was given by T.S. Sadasivam, I think. So the example of vivotoxin is fusioric acid, and the best example of pathotoxin, so which plays a major role in pathogenicity. Pathotoxin best example is pyrrolidine, which is produced by pyrrolidine virus, which causes rice blast disease. The next one is the elicitor can determines the pathogen. So resistance. So elicitor actually determines what? Resistance, virulence, avirulence, all the above. Generally, this elicitor determines the avirulence. Okay. So which of the following substance involved in the production of plant diseases? Enzymes, toxins, growth regulators, all the above. Generally all the above, because toxins, enzymes, growth regulators and other kinds of, one more thing is toxins, enzymes, growth regulators and polysaccharides as well, all are involved in the plant disease. For example, not all pathogens will produce toxins. Some pathogens maybe produces enzymes. That enzymes also act as a toxin. This growth regulators also act as a toxin. For example, if you take Agrobacterium tumefaciens, so due to hypertrophy and hyperplasia, the disease will come. How this hypertrophy hyperplasia occurs? Due to the production of growth regulators, right? So that is how it will act. So all are indirectly or directly causing the diseases, these toxins, enzymes, growth regulators are. So the answer is all the above. So which plant pathogen attack diverse host plant? I repeat, which plant pathogen attacks diverse host plant? Autotrophs, biotropes, necrotropes, all the above. So this generally this necrotropes can affect diverse or wide range of croplands. So which gene of rice blast, they are asking, rice blast encodes a protein that enables the pathogen to recognize the formation of upper story in the host surface. So due to the presence of which gene, the plant can easily recognize the upper story formation by the rice blast. PTH gene is actually responsible and identifies this formation of upper story on the host plant. So which of the following are antimicrobial substances that are produced by the plant and accumulates at incompatible pathogen infection? I repeat, antimicrobial substances produced by the plant and accumulated at incompatible pathogen interaction. In the previous slide only I told you phytoalexins. Phytoalexins are the plant protection substances which are produced by the plant after infection. So at the incompatible reaction to protect the host for further spread of the infection or further invasion, they produce phytoalexins, that is called during incompatible reaction. Various kinds of phytoalexins are there, but the first discovered phytoalexin is P-satin, which is produced by the P. So first step of activation of defense responses against the invading pathogen: signaling, elicitation, recognition, infection. The first step is, first of all, we need to recognize the the presence or the entry of a thief. The thief in the sense these substances which are produced by the pathogen. So once you recognize, then only you can act according, right? So the first step is the recognition. The term phytoanticipants was coined by, I repeat, not phytoalexins, phytoanticipants. K.O. Muller, O.A. Dexter, Mansfield, and Borger. So generally, the term phytoanticipants was given by Mansfield. Low molecular weight antimicrobial compounds which are present in the plants before infection of microorganisms. Phytoalexins are generally produced after infection, but this substances which substances will produce before infection is a phytoanticipants which actually produce before infection. Virulence factor of Ralstonia solanacearum, which generally causes bacterial wilts or bacterial blights, causing the bacterial wilt or bacterial blights of solanaceous crops, especially solanaceae, potato, tomato, especially solanaceous kind of crops. So what is the virulence factor of this Ralstonia solanacearum which helps to cause the disease? So EPS1, EPS2, EPS3, EPS4. So exopolysaccharides, EPS1 I think. So the EPS1 act as a virulence factor for this Ralstonia solanacearum. There are races also there, 1, 2, 3, 4, something like that. So failure of xylem water translocation is actually caused by dieback, gummosis, vascular wilts, all the above. So generally, most importantly, this vascular wilt blocks the xylem and phloem vessel, so there will be no translocation of water. So generally, vascular wilts. Sequence of events in pathogenesis. So in pathogenesis or occurrence of disease cycle are causing the disease, what are all the sequences? So attachment, penetration, host recognition, infection, invasion, all those things that we have given here. But I'm telling the answer. So here, first of all, the pathogen need to come and attach. The next step is penetration, host recognition, infection, invasion, colonization. This is actually the sequence or order. The next one is Victorian toxin is produced by. Victorian is a toxin which is produced by Helminthosporium carbonum, Cochleobolum carbonum, Helminthosporium victoria, all the above. So it ends with Victoria, right? So Helminthosporium victoriae, which is produces this Victorian toxin. I think it's a world's host specific toxin. I think something so. So world's first host specific toxin is this Victorian, probably produced by Helminthosporium victoriae. Which one of the following is a non-host specific toxin? Which is a non-host, not host specific, non-host. So again, in toxins also two kinds will be there, host specific and non-host specific, that we have already explained in the previously published videos in physiological and molecular plant pathology in the previous year. So please go through so that you will have a little idea on this. So which of the following is a non-host specific toxin? Fumaric acid, oxalic acid, PC toxin, both A and B. So the answer is both A and B, that is fumaric acid and oxalic acid are the best example for this non-host specific toxin or host non-specific toxins. Mode of action of wildfire toxin. So wildfire toxin is again the first purified toxin probably that is produced by Pseudomonas syringae, Pathovirus syringae. So what is the mode of action? Inhibits glutamine synthesis, affects the membrane permeability, inhibits the cell wall synthesis and the lipid biosynthesis. So this mode of actions also very important, sometimes they will ask. So the best this wildfire toxin is inhibits the glutamine synthetase activity, that actually inhibits this glutamine synthetase activity. So PR proteins, PR5 encodes or codes. This is a very important one. PR1 is beta 1, 3 glucanases, తర్వాత antifungal, antibacterial, so many things. This PR5 is actually thamatin-like protein or a tramatin-like protein we used to call, but PR5 encodes tramatin-like protein. Elicitors are produced by host, pathogen, both A and B, none of the above. Generally, elicitor is the product or substance or a kind of stuff which is produced by pathogen. In response to that, the host will recognizes or receptors. It recognizes, this receptor identifies and recognizes. Okay, so this elicitors are the pathogen origin. Okay. Enzymes are responsible for disintegration of host cells. Disintegration or lysis, which actually digests the cell wall. Cellulosic enzymes, pectinolytic enzymes, both A and B, and none of the above. So both cellulolytic and pectinolytic enzymes, which actually degrade the tissue, are tissue disintegration enzymes. Okay, these pectinolytic enzymes we mainly observe in soft rot of carrot. Right, pectinolytic enzymes, the only that they are degrading. Right. Next one, pectinolytic enzymes are produced which of the following pathogen? Just now I told you soft rot of carrot, Irvinia carotovora, right? Pythium, Phytophthora, Irvinia carotovora, all the above. Generally, this Irvinia Keratovera produces these pectinolytic enzymes, but it is also, like you can see, softening. Like if you see this pythium infected plants, you can see some kind of softening or disintegration of tissues also. So Phytophthora also can do the same thing. So this answer is all the above, but there is no pythium and Phytophthora. You can go with this Irvinia keratovera, which causes soft rot of carrot. Irvinia keratovera subspecies keratovera. Which of the following structure is produced by some fungi to help them to penetrate the epidermal cells? Which means there, what is the structure which actually helps to penetrate into the epidermal cells? Upper sorium is generally a attachment organ, right? Germ tube, promycelium, conidia. Conidia is like asexual spores which are produced by Ascomycota and Basidiomycota group of fungi. Generally, if you see, this aprosorium not only attaches and also helps to penetrate inside. So the main culprit is here aprosorium. So germination peg will be there. The germination tube, which means actually directly germinating from the spore, that is germination tube. From aprosorium, a small tube penetrate inside, that is called penetration peg. So since there is no penetration peg here, so the answer is aprosorium, because this penetration peg is actually developed from this aprosorium only. And the last one is, which of the following signal pathways signaled the pathogenic fungi? So what are the signal pathways? CAMP, MAP, MAPK, both A and B, none of the above. So both A and B, CAMP, cyclic adenosine monophosphate pathway, and mitogen activated protein kinase are the both the pathways which are responsible for this signal transduction, signal exchanges. Okay. So for regarding this CAMP and MAP, especially on molecular plant pathology, we are planning to do in separate videos. So these are all the answer for references. So for more information, students can refer this book, A Vision into Plant Pathology, complete student version. For further doubts, clarification, student can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.